you have any questions, and Dr. Staudinger will help direct those questions to Euna. So to get started, first, it's my pleasure to introduce Ms. Euna Kim. She came to KCU with the Bachelor of Science in Human Biology from the University of California, San Diego, where she spent a semester studying biology abroad at Yonsei University in Korea. Her research background comes from working with dendritic cells and determining the role of CAMP in CD4 T Cell differentiation. Since coming to KCU, Euna has authored an article with Dr. Arnce titled "Warmer Weather Brings Ticks, Prepare for Tick Season in the 4 States," which was published in Show Me the Ozarks magazine. And more recently, she's helping to compile the COVID-19 resource document for KCU students, faculty, and staff that can be found on the Canvas page. And today, Euna will be presenting on medications and treatment of COVID-19. Euna, take it away.
Thank you so much, Shelley. Let me start sharing my screen. Okay, so I am presenting today on promising medication trials for the treatment of COVID-19. And as Shelley said, my name is Euna Kim, and I'm a 3rd year at KCU. So there has been a lot of interest when it comes to medication for treating COVID-19. Especially there's been a ton of interest into different antivirals as well as immunomodulators. Some of these that have been really prominent in the media are hydroxychloroquine, interferon beta, lopinavir, ritonavir, favipiravir, just to name a few of them. The 2 that I'm going to talk about today are remdesivir and tocilizumab, and they have been showing some pretty promising results. Before the remdesivir article came out, there weren't that many therapeutics that were showing good results to fight COVID infection. So just a slight introduction on them. Remdesivir is a viral RNA polymerase inhibitor, so it's a nucleotide analog, and it was shown in the past to be effective against SARS-CoV-1 as well as MERS. And then recently, it's been shown to be effective against SARS-CoV-2 in vitro. SARS-CoV-2 is the name for the virus that causes COVID-19 infection. And because of that, there was a lot of anticipation for the drug remdesivir. And this drug was developed and is owned by Gilead Sciences. And currently, it's not used for anything. It was first formulated to fight against Ebola, I believe. But it wasn't effective against that, and right now it's not currently being used for any disease processes or to fight against any viruses. And the FDA has filed an emergency use authorization for remdesivir so that it can be used by physicians compassionately in COVID-19 infections. So tocilizumab is an interesting one. It's an IL-6 receptor inhibitor, so it binds to the IL-6 receptor. IL-6, as well as GM-CSF, and GM-CSF stands for granulocyte macrophage colony stimulating factor. Those 2 cytokines have been shown to contribute a lot to the cytokine storm that causes a large amount of damage in COVID-19, especially ARDS, the acute respiratory distress syndrome, the inflammation that happens in the lungs. So because they're such major players, it was theorized that knocking out that IL-6 inflammatory pathway would decrease a lot of the inflammation and really help in mitigating the negative effects of this infection. Tocilizumab is developed and owned by Roche, and it's currently used to treat rheumatoid arthritis as well as some other autoimmune diseases. So I'm going to go over the remdesivir article first, and this is a little bit on remdesivir's mechanism of action. It was a repurposed drug, I said, that was first developed for Ebola, but how it works is that the SARS-CoV-2 virus, it is a single-stranded positive sense RNA virus. So once that RNA gets deposited into our cells, as you can see, it uses the ACE2 receptor to enter into our cells. That RNA is translated into the viral polymerase, so the RDRP. Oh, wait 1 second. I need to use my insert pointer. Give me 1 moment, please. Yeah, it's translated into the viral polymerase protein. And then once that happens, this drug, remdesivir, interferes with the actions of the viral polymerase protein, thus preventing transcription of a lot of the downstream RNA that will make the rest of the viral RNA proteins. So that's a little bit of overview on that. So this study, remdesivir for the treatment of COVID-19, the preliminary report, it was published on May 22nd, 2020, so pretty recently. It was a randomized controlled trial, double-blinded, and there were about 60 trial sites worldwide, and the randomization was done by study site as well as disease severity. So there were 1,059 participants in this trial. 538 of them were in the remdesivir group, and 521 of them were in the placebo group. The dosages that they received of remdesivir was 200 mg on the first day, and then 100 mg every day after until day 10. And the placebo was given in the same pattern and volume as remdesivir. And all of these patients received supportive care, and they were all assessed for a full 29 days. So this is some of the demographics of that patient population. They were about 58.9 years old. That was the median age. Most of them were male, but not an overwhelming amount. 64.3% of them were male. And this, as you can see here, is the median time of onset. So it was 9 days. And this meant that most of the patients had symptoms for 9 days before they were entered into this trial and given remdesivir and or placebo. A majority of them had comorbid conditions. In fact, 52.1% of them had 2 or more coexisting conditions, the most common one being hypertension. And this ordinal scale down here, I will discuss in my next slide. One more thing. So 88.7% of these patients that were enrolled in this study had severe disease at enrollment. And severe disease was defined as needing ventilation, whether it was invasive or non-invasive, needing supplemental oxygen, having an O2 stat less than 94%, or having tachypnea, so more than 24 breaths per minute. So this is the clinical scale that was used to assess their status. There were 8 categories in it. All of the patients that were enrolled were in groups 4, 5, 6, and 7. Groups 1, 2, and 3 were considered to be the recovery group. So as soon as any of the patients moved from a higher score to either 1, 2, or 3, they were considered to be recovered. A majority of the patients, not a majority of the patients, the largest sample size was in group 5, which were hospitalized patients that required any supplemental oxygen at all. Okay. These are the Kaplan-Meier plots of the outcomes. And panel A shows the overall outcomes. So that's of everyone that was enrolled. The other panels, B through E, show the outcomes based on their baseline ordinal score, their baseline clinical status. So I'll go over panel A first. So overall, the remdesivir group, as you can see here, approached about 75% recovery rate. And compared to the placebo group, they approached at about 60% recovery rate overall. In panel B, now we're looking at the patients that were the least severe patients, which were the patients not receiving oxygen, but they were hospitalized. So the remdesivir group, pretty much 100% of them recovered, and the placebo group, about 85% of these patients finally recovered at the end of the 29 days. So this plots their progress out all the way to 29 days. So these are panels C, D, and E. So these are groups 5, so patients who are hospitalized and receiving oxygen. 6, patients that are hospitalized receiving high flow oxygen or non-invasive ventilation. And 7, the worst off of the patients, which were patients that were receiving mechanical ventilation, invasive. So as you can see here in panel C, The remdesivir group, in this group, had about an 85% recovery rate, and the placebo group had less than 75% recovery rate. And this group actually showed the most difference in effect compared to comparing remdesivir to placebo. They had the greatest beneficial effect, or so it seems. In panel D, now we're looking at the worse off patients, and the placebo and remdesivir groups get closer and closer in terms of recovery time and clinical status. So only about 50%, maybe about 60, 55% of these patients recovered in both of these groups. And then as soon as we move to E, it's apparent that the remdesivir group didn't benefit very much compared to the placebo group. And in fact, it actually looks like the placebo group did a little bit better. So the placebo is the red line. And it looks like they might have fared a little bit better. And in kind of thinking over this, it makes sense because the patients that were worse off later, there was less effect to using an antiviral that would interfere with the virus proliferating much later in their course of disease. And I don't believe that the difference between these two groups right here in panel E are significant. So it's a bit of an insignificant difference. This chart looks at the outcomes overall, as well as broken up by the ordinal score, their clinical status. So overall, let's look at this first. The remdesivir group had a median time to recovery of 11 days, compared to the placebo group, which had a median time to recovery of 15. So that's pretty significant. If you look at the rate ratio between these two groups, it's 1.32. What this means is that the remdesivir group had a 1.32% time, they were 1.32 times more likely to recover compared to the placebo group. And this was clinically significant. The hazard ratio was 0.7, and what this would mean is that the remdesivir group had a 30% in risk reduction compared to the placebo group. However, this was not significant because the confidence interval crossed the null value, which was 1. But I thought that was interesting anyways. The rest of these break up the patients by their baseline ordinal score, 4, 5, 6, 7. And all I can say is that the worse the baseline ordinal score was, the less difference there was seen between the two groups. There were some adverse events that were associated with remdesivir as well as placebo. There were about 114 patients in the remdesivir group who said that they experienced an adverse event, and 141 in the placebo group that said that they experienced an adverse event. However, after looking at their symptoms, it was decided that only 2 from the remdesivir group and 2 from the placebo group actually experienced adverse outcomes due to those drugs or placebo. But the most common reported adverse event was respiratory distress, which is also a common part of the disease process of COVID infection. So it's very likely that the disease progression itself caused these adverse events, and it wasn't really related to remdesivir or placebo. So the conclusions of this study is that a 10-day course of remdesivir is superior to placebo in promoting recovery from severe COVID-19 infection. And they concluded this by looking at the median recovery time, remdesivir of 11 days and placebo of 15 days. Also, the largest benefit was seen to the baseline ordinal score of 5, and that group was the group that was hospitalized and receiving any oxygen at all. And so their groups had the biggest difference that was seen, but also the largest sample size was in that group. So that could also contribute to that information. To that outcome, sorry. This article also concluded that there was a high mortality despite remdesivir use. And so it seems like an antiviral alone is not enough to fight against severe COVID infection. More therapeutics in combination are required to treat the inflammation that is present in the patients that have already undergone a significant disease process. Just as a side note, a week after this article was published, there was another article that looked at remdesivir treatment for 5 days versus 10 days. And that study found that those two treatment cycles produced the same outcome. So 5 versus 10, the same clinical outcome.
Student Dr. Kim?
Yeah.
We have a question from Student Dr. Volker.
Okay.
Hi, can you hear me over there?
Yeah.
Yes.
So I had a quick follow-up question with the conclusions here. Do you know if this study group has planned a follow-up to see viral shedding after the course of remdesivir is over, to see if that changes? And theoretically it does, looking at the data, but I didn't see anything in the paper about that.
I don't think I've seen anything related to that or answering your question, but I could be wrong. Has anyone else heard anything?
Yeah, I've had conversations with numerous folks, and what they're calling it is viral remnants are detected weeks, even months after in some patients. Darren, you and I had a conversation about that. Would you care to chime in on this, Dr. D'Agostino?
Sure. So this is one of the areas that's being misinterpreted in the media and social media. The viral remnants are essentially pieces of RNA that are picked up by a very sensitive tool, the PCR testing that's done. And in some cases has been misinterpreted as virus. But when you really look at this, what's happening is cultures are not growing because the virus is actually not there. These can go out to upwards of 80 days in some patients. Now, those are not a lot of them, but it can be very long. The challenge with this, really what happens at that point is these viral remnants, carcasses, I call them, basically are evidence that there was an infection. They're not infectious anymore, but can also be used for surveillance in a lot of different places, because if you do find the viral remnants, the only way to have it is to have had the virus. These do not equate to an immune response, so we don't know if people are no longer infectious. We don't know if people are immune at that point, not infectious, but immune, because that's all based on your immune response, and the outcome of that immune response is still pending. We don't know the true immune response and protection we have. So those remnants have to be, you have to take that into account, and that's a clinical scenario. So a person's not sick anymore, but you're finding those particles that are testing positive. You have to, as a physician, interpret that as they're not sick anymore, and more than likely these are remnants.
Okay, thank you.
Thank you.
Devin, does that answer your question?
Yes, it does.
Okay. And that was my last slide. So if there's any questions.
Student Dr. Kashim?
Yes,
Darren?
hi. Yeah,
Hi.
hi, Una. So you were talking about the 5-day course remdesivir article. Was that sample size bigger than this article?
No, it was not. It was actually significantly smaller than this article. I think, I could be wrong, but I think it was only a few hundred patients that they looked at for that article.
Okay, and would you, I'm not sure if maybe there were more adverse events compared to the 5-day versus 10-day course, but would you recommend a 5-day or a 10-day course of remdesivir?
So the other article where they talk about that, I believe they didn't find a significant difference in adverse events, and the clinical outcomes that were derived from those two courses, they were very similar. And I think What else was I going to say? Anyways, I think as a physician, if I had to choose, I would probably go with the 5-day course, seeing as the other articles seem to have some pretty compelling evidence that it would be a similar outcome.
Okay, thank you.
Shelley Boyer has a question.
Hi, Una. Thank you for the presentation. I was just wondering, and I may have missed it, it could have been on the slide, one of the tables, but did they track the days of onset of symptoms before administering the drug or placebo? Meaning, like, I'm just wondering if progression before treatment was administered was because of the unwillingness of the patient to go to the hospital within, that earlier time.
Mm-hmm. So so these patients did have symptoms for 9 days, but I'm not sure if any data was collected on them during that time. Is that what you were asking, Shelley?
Yeah, like if they were all administered the drug after 9 days of the symptom onset, or if it was just 9 days after testing positive, where that 9-day kind of come from, if that was equal across the board, I guess.
So it wasn't equal across the board. I think that was just the median time, but it was from symptom onset. So ever since they started complaining of any symptoms that was possibly COVID related, that's when they started measuring that outcome for that.
Okay. Perfect. That answers me. Thank you.
Thank you.
We have another article. Does anybody else have any questions before we move to the next article? Okay, Una, take it away.
Okay, so my next article is about tocilizumab. It's also called Actemra. It's owned by Roche Sciences. So what tocilizumab does is that it attaches to the IL-6 receptor. So there's both a soluble receptor as well as a membrane-bound receptor. And to my knowledge, they have the same structure, and the only difference is in the transmembrane region. But they set off a similar cascade of events, which is increasing the inflammatory response. So everything that you see down here are all ways that IL-6 increases the inflammatory response in the body. Okay. So the article title is Effective Treatment of Severe COVID-19 Patients with Tocilizumab. This was published on April 29, 2020, which was kind of a while ago if you're looking at research regarding COVID-19. However, this is one of the better articles on this drug, so I chose this one. So this was not a randomized controlled trial. There were only 21 patients and there was no placebo arm. So these results have to be taken with a grain of salt. So these 21 patients were chosen from 2 hospitals in China, and they had to have been diagnosed with severe or critical COVID when they were chosen. 18 of them received tocilizumab once, and then 3 of them developed a fever within 12 hours of the first dosage, so they received a second dosage of tocilizumab. And if you're interested in the dosage, it was about 4 to 8 mg per kg, from a minimum of 400 mg to a max of 800 mg. The second dosage that those 3 patients received was the same as the first dosage they had received. As you can see here, these are some demographic characteristics of the patients. Median age of 56.8. But I would like to point out that they had a very large age range here, from 25 to 88. And also an overwhelming amount of them, of the 21, were male. So 85.7% of them were male, and 3 of them were female. So this table looks at laboratory tests before and after tocilizumab administration. So some of the main lab results, or some of the main laboratory values that are associated with severe COVID infection, are a normal white cell count, lymphopenia, and increased CRP. The lymphopenia, several studies have shown that the more severe the degree of lymphopenia, the worse the infection is. So if you look right here, the lymphocyte percentage before tocilizumab administration was 15.52. 5 days after administration of this, the lymphocyte percentage went up 22.62. So the lymphopenia improved. The CRP value of these patients before tocilizumab was 75.06. And then after 5 days of administration, it fell dramatically to 2.72. IL-6 is an interesting value right here. It was at 153.44 before administration. 5 days after, it was at 274.90. And this increase is actually to be expected, and it makes sense because tocilizumab attaches to the IL-6 receptor. So all of the IL-6 that is continually being made is kind of free-floating in the serum. So there's a higher concentration of them because they've been displaced. And I don't remember if I've mentioned this, but CRP is an acute phase reactant that is downstream of IL-6. Oh, actually, before that, one thing that I found interesting about this lymphopenia is that their theory on why severe COVID infection causes more severe lymphopenia is that there's tissue sequestration into the tissues. So the lymphocytes are sequestered into the tissues. Sorry, I said that confusingly. So these are some CT scans of patients before and after tocilizumab administration. A and D are the same patient, B and E are the same, and C and F are the same. Let me just give you a really quick primer on how to look at these. So imagine as if in front of you there is a patient on their back lying on a table and their feet are facing towards you. So you're looking up through them and there's a cross section that you're looking at. So this is the patient's spine. So this is their back. This is their front. This cross section right here is looking at the lungs on either side as well as the heart in the middle. And this is the right hand lung of the patient. This is the left hand lung. This right here are some pulmonary markings that are actually rather normal. So this is what the lung should look like, some small markings that are there. So this patient, before tocilizumab administration, you can see that there's quite a bit of consolidation in the back part of the lungs on both sides. 1 week after tocilizumab administration, a lot of this consolidation is slowly breaking up and going away on both sides. In this patient, on the right-hand side, their lung is severely inflamed. There's inflammatory debris throughout most of their right-hand lung. And then 1 week after, a lot of this debris is lightening up and being absorbed, reabsorbed. And in this last patient, again, you can see that there is a mild amount of inflammation in the back part of their lungs on either side. And then 1 week after administration, some of that is breaking up and becoming less consolidated. So they found that 90% of the patients, I think 18 out of 21 of the patients showed CT scan improvement 1 week after tocilizumab administration. These panels show the levels of inflammatory markers after tocilizumab administration. So this one is CRP. Oh, sorry, this one is temperature. This one is CRP. So all of these patients are graphed out right here. And as you can see, by day 5, most of them have normalized in CRP levels. For temperature, after 1 dose of tocilizumab, all of the patients dropped in temperature, and 18 out of 21 of those patients were at a normal temperature after 1 dose, after 1 day of tocilizumab. And their temperature stayed relatively stable throughout the next 5 days. In panel C, this is the oxygen concentration that was needed after tocilizumab. So before administration, you can see that the patients are pretty much all over the spectrum in what degree of oxygen concentration that they need. And then after administration, very quickly, you can see that all of the values start to consolidate. So by the end of the 5th day, all of the patients needed less than 60% oxygen concentration. And in fact, one of the patients didn't need oxygen at all. They're graphed at the 21%, which is room air. oxygen concentration in room air. And in panel D, this is the resting oxygen stat after tocilizumab administration. And again, before administration, their O2 stats are all over the spectrum from 100 to 90. And then 5 days after administration, it consolidates, and all of them are at an O2, resting O2 stat of 96% or above. So what are the conclusions of this article? The conclusions are that tocilizumab administration did improve clinical symptoms within 5 days of administration, and they did decrease the laboratory values that are associated with severe COVID-19 infection. There is also a very favorable safety profile to this drug. However, as mentioned before, the study is very low powered because there were only 21 patients and there was no placebo group. So more randomized control trials are needed to really show the clinical benefit of tocilizumab. Yes, as a side note, also, Roche owns tocilizumab, Gilead Sciences owns remdesivir, and they are going to partner up soon and see if the remdesivir and tocilizumab combination is actually really effective or not in treating patients with severe COVID-19 infection. And Roche is also planning on doing much bigger trials with just tocilizumab to kind of solidify its clinical efficacy. And actually, many different countries right now are doing trials on tocilizumab. There's other IL-6 receptor inhibitors that are being looked at too, such as sarilumab and siltuximab, but tocilizumab is the main IL-6 receptor inhibitor that is being studied. Any questions?
Any questions for our speaker? Let's see, who is that? Give me a second. Shelley.
Hi, Una. Again, thank you for the presentation. I was wondering with some of the numbers on one of the tables, I think it might have been like the first or second table.
This one?
Go back.
This one?
Well, maybe it was the very first one. Keep going.
I think this is the first table. This is the second one.
Okay, maybe. Where did I see the numbers that started with the number of participants? Maybe this was the smaller study, wasn't it?
Mm-hmm.
Okay.
Yeah, the 21 patients.
Yeah, I don't have a question. I just needed to go back to the slide so I could look at the data.
Oh, okay. Yeah, please.
Thank you. Sorry. Thank you.
No, not at all.
Student Dr. Kashim.
I'm not sure if you spoke about it in your presentation, but what was the standard of care that these patients were, being treated with before being given tocilizumab?
Yeah, sorry, I did mean to mention that. Sorry. This is the last slide. So I don't know if I mentioned this, but all 21 of these patients were discharged 2 weeks after tocilizumab administration. And the standard of care that they were given along with tocilizumab were lopinavir, ritonavir, as well as methylprednisolone and interferon alpha, I believe. So those 3 drugs were given along with tocilizumab.
Thank you.
Mm-hmm.
Any other questions? Okay, then what we should do now at this point is thank our Journal Club speaker. So everybody unmute and clap. Thank you.
Thanks for listening to me.
A couple of announcements real quick, and I'm going to share my screen very briefly if I could. Hopefully you guys are seeing this. I don't see you guys anymore, so I need to hear that I'm sharing my
Yes.
screen.
Mm-hmm.
Okay, good. This is the MKRC homepage. There's a research spotlight, a summer seminar series, which is weekly on Thursdays, and then here's the weekly on Friday link. If you click on this, the other day I uploaded the student doctor's COVID-19 resource document. So all you need to do is merely click here on the blue lettering, and you'll download this document, and then you can open it. It's downloaded as a PDF document. I can make this a little bit bigger. Here are all the student doctors that contributed to this document, as well as Haley Neiman, the summer student who's working with me virtually from Bates University. So grateful to the student doctors for taking Haley under their wing, and she's considering medical school, and we're trying to recruit her to KCU. But at any rate, the table of contents is all clickable, and the table of contents is enormous. And so you don't have to read all of this 195-page document, but there are parts of it that you may be interested in, and the table of contents are all clickable, and that'll take you to that part of the document that addresses this. And then there are embedded links that take you to the actual website where that's being referenced in the COVID-19 document. So I just wanted to take that moment to orient you folks in this room to that notion. And that's, again, available at the MKRC Canvas site. Once again, I appreciate so much the effort that you student doctors have gone to produce this. This is an amazing piece of work, and I'm honored to be involved in facilitating your efforts and guiding you a little bit in that regard. So thank you so much for this. And I'm going to turn it over to Shelley to wrap things up and talk about the upcoming talks we have. Ms. Boyer?
Thank you, Dr. Staudinger. And again, thank you everybody for coming. And hopefully you'll find your way back here every Friday from 1 to 2 this summer. And if you're available on Thursdays for the seminars as well. Next week, we're going to have the topic of socioeconomic disparities and deaths in the U.S. And student Dr. Stephanie Bates will be delivering that for us. So thank you again, everybody, for coming, and have a nice weekend.
Thank you.